Is there any soybean oil in your house?
No.
Canola?
No.
Corn?
No.
Is there any seed oil in your house, Tommy Wood?
I don't think. There's olive oil.
That's not it. That's a fruit oil, right?
Yeah, I know.
What's up, guys? Welcome to this week's podcast with my friend Tommy Wood. He is probably one of the smartest humans I've ever met. So it was a real joy to get to talk to him this week about all kinds of things. Our conversation is wide-ranging. The main focus of our conversation is on dementia, Tommy's thoughts on dementia. Tommy is a neuroscience, neurobiology, developmental neuroscientist at the University of Washington. He works on how to help grow and repair infant brains when they've been injured by all sorts of insults. So he thinks a lot about the brain. We also talk about lipids, LDL. We talk about glucose measurements, whether continuous glucose monitors are valuable, whether you should worry about an absolute threshold for your glucose. We talk about seed oils a little bit. Tommy is a little, I think, conservative in this podcast in his perspective, but he's accurate in his portrayal of the fact that the seed oil literature needs to be made more robust by further experiments. But I'm going to have Tucker Goodrich and Jeff Nobbs on the podcast soon to do a little more on the seed oil front and really go down that rabbit hole in a deeper way. I think despite the fact that we can't really come to any major conclusions about seed oils in this podcast with Tommy, you'll note that Tommy has no seed oils in his house, and I don't think he's a fan of them, though I don't want to put words in his mouth. In fact, I know he's not a fan of them, but we need more good science there. So enjoy this podcast with Tommy Wood on dementia and his unique perspective on one of the things that we are doing or not doing as a human people in 2022 that could be contributing to our progression toward cognitive decline in the middle and the end of our lives. So, all right. Also want to let you guys know, before we move on, the Kale is Bullshit shirts have arrived. You can go to kaleisbullshit.shop to get your Kale is Bullshit shirt, to get your seed oils or bullshit shirt. We've got hats, we've got hoodies, got all kinds of good stuff there. If you want to support the movement, be a part of the tribe, I can't wait to see some of you guys in these shirts. I'm going to be back in Texas in mid-November in Austin, and I'm going to be at those Whole Foods calling bullshit. You know it, and I hope I see you there, guys. On to the podcast with Tommy Wood. Enjoy this one. Tommy Wood, thanks for coming back on the podcast, my friend.
Thanks for having me back. It's been a while.
It's been way too long, man. It's been way too long. I'm now in Costa Rica, where Tommy has a crazy story of an encounter with a viper of sorts. Maybe we'll save that for later in the podcast. Tommy is still in Seattle, but Tommy, this is your third appearance on the podcast, I think, separated by at least a couple of years. We've done previous episodes on ApoE4, which is something that I'm sure we'll talk about a little bit in this podcast. And then I think in our first podcast, we kind of touched on the blue zones. That's something we'll probably rehash in this podcast as well. But what are you doing out there in Seattle? Tell people a little about your background if they don't know who you are.
Yeah, sure. So I'm an assistant professor of pediatrics and neuroscience at the University of Washington. Most of my job is trying to find ways to treat babies with brain injury. But I do more and more work in other kinds of brain injury as well. I have some experience on the clinical side and the applied side with traumatic brain injury and cognitive decline, Alzheimer's disease, but just starting to do some of that work in the lab as well, both trying to better understand the disease pathology, but then also ways to treat it. So increasingly, I'm sort of putting together this idea that, you know, you only have one brain and you have to look after it for your entire lifespan, and things that happen at different stages will then affect what happens later on as well. And that comes down to, you know, what your brain's been exposed to, nutrition, sleep, genetics to a certain extent, you know, all that kind of stuff kind of comes together. And it's important, you know, regardless of how old you are. So how can we sort of tie those things, you know, across the entire life and make sure that our brain is as healthy and happy as possible for as long as possible?
Yeah, I think this will be a fun episode talking about brain health and Alzheimer's. I know you published a recent paper with Josh Turkel that I'm excited to tell people about and have you share about. Critical question as we start. Have you ever eaten brains?
Yeah. Yes, I have. I think most recently goat brain.
All right.
I included it as part of like a pate type thing. But then also I've had some desiccated brain in capsules from a company that will remain nameless.
I might know the company. There's actually some interesting evidence about desiccated brain and derivatives from desiccated brain, aren't there? I mean, aren't there studies? I remember looking at studies on, what am I thinking of? Phosphatidylserine from bovine cortex that's been used in cognitive models of decline.
Yeah, so I think the best evidence we have sort of at the human level is for phosphatidylserine as almost like an adaptogen. So basically modulating cortisol levels, particularly if cortisol levels are high, sort of bringing them down, and maybe potentially improving sleep and sort of anxiety, stress around that. What's interesting is that there was obviously a big deal with BSE, bovine spongiform encephalopathy, particularly in the UK and Europe back in the 90s. That's actually the reason why I can't give blood in the US, is because they assume that I have been contaminated with BSE, having eaten beef in the UK 30 years ago. But because of that, brain supplements became, or eating brain was banned, or was certainly not available for long periods of time. And when they redid some of the studies with looking at phosphatidylserine, but looking at it from a plant derivative, I think you can get it from lecithin, so plant lecithin. It didn't have the same effects. So it may not actually be the phosphatidylserine. It may have been whatever else was coming from the bovine brain as part of the supplements that was coming along for the ride, or maybe there was some kind of like synergistic effect. So that's certainly interesting.
Yeah, when I was in Tanzania and I was telling my friends that I ate baboon brain, they were all convinced that I was going to get some sort of prion disease, and so I guess that might explain some things. Who knows if I have prion disease now? I don't know. And I remember my residency, when I was on my neurology rotation, there was a resident talking about looking for 14-3-3 protein, I think, in the CSF of a patient. And he was doing the spinal tap, and a little bit of the spinal fluid hit him in the eye. And he was just so scared that he was going to get Crutchfeld-Jakob or some other sort of prion disease. Since we're talking about prions, let's tell people what prions are, and then we'll get into Alzheimer's. It's all brain stuff today, guys. You'll get the idea.
Yeah, I mean, so there's this group of prion-based diseases, and they differ from species to species, but there seems to be some evidence that they can kind of, they're zoonotic, right? They can transmit from one to the other. So we would call it Creutzfeldt-Jakob disease in humans, BSE in cows, Scrapie in sheep, and the idea is essentially this almost like pathogenic misfolded protein, which can sort of like propagate. And associated with that, you get all kinds of motor, cognitive symptoms, and then eventually early death. And there's no treatment as far as we know. And there's been a whole ton of sort of investigations trying to figure out what causes them, like what might initiate the misfolding and sort of propagation of these proteins. Like, is it, are they actually just like viral bodies that can be transmitted in that way? Are they? Is it due to like different micronutrients? So there were some. There was some people who thought that maybe like an imbalance between like the availability of copper and zinc may cause the proteins to be folded differently, and that may cause it. I don't think anybody really knows, or if they do know, that's not something that I've read in the literature recently. But because BSE has sort of not been a thing that we've really seen much recently, I think they've sort of there's been less interest in it, but still certainly something that people have been concerned about. And there was a whole period of time where they thought that, so I think there were some patients with CJD underwent neurosurgery, and then the tools that we use in neurosurgery were improperly autoclaved, and then it was transmitted to other patients. So certainly seems to act, at least in some ways, as like a transmissible virus like response almost. But what sort of initiates it, I think, is still incompletely understood.
It's a scary thing for sure, isn't it? I mean, Kuru is essentially like this, right? And it was a tribe in Africa, I believe, that was practicing some sort of cannibalization. And wasn't the idea that there was some spontaneous mutation that caused a Crutchfeld-Jakob type prion disease in humans, and they ate that person? Wasn't Kuru like spontaneous that just arose in the population?
I think so. I mean, it appeared due to cannibalism of other humans' brains. But yeah, exactly how it was initiated, I don't know. And It could again be similar to some of the other things that people have thought about in terms of, nutrient availability, whether, metals are causing differential folding of proteins. And then once it's started, it sort of self-propagates. But how it initially came up in that setting, we would call it spontaneous, but there's probably a biological reason for it. We just maybe don't know exactly what it was.
Yeah. So, okay, prion diseases aside, they're much more rare than dementia. Let's talk
very rare.
Very, very rare, thankfully. Hey guys, I thought this was the perfect moment to just pause this podcast with Tommy for one moment to tell you about Mood Memory and Brain from Heart and Soil Supplements. This one just came back into stock. And I thought it was so cool that in this podcast, Tommy and I are talking about the benefits of desiccated brain. And I love when Tommy mentions, when you look at phosphatidylserine from plant foods, it did not have the same benefits as desiccated brain from cows in terms of memory and cognition in elderly individuals, which is why I love this supplement. I love Mood Memory and Brain from Heart and Soil. It's now back in stock. And I want to read you another review from Betty O, a verified buyer of Heart and Soil. She says, I started taking Mood Memory and Brain in hopes that it would help with my fibro fog. Trust me, that is a real thing. I'm working full time. I was really struggling with it on the job. I'm happy to say this supplement is working wonders. And it helps me so much. My memory is clear and so much better. My head feels more clear as well. Thank you for making these great natural supplements that help with so many things. I am definitely a loyal customer. You can find Mood Memory and Brain and all of our supplements from Heart and Soil. They're all grass-fed, grass-finished, regeneratively raised, all from New Zealand sourced at heartandsoil.co, heartandsoil.co. And this one, Mood Memory and Brain with desiccated brain, is back in stock. So if you need some brain assistance, some memory benefits, if you know someone in your life who has declining memory, check it out. Get some desiccated brain in their life, and I believe it'll help. It's a powerful thing. It's been studied in research. Back to the podcast with Tommy Wood on dementia. Check us out at heartandsoil.co to get some of this. I don't think I have it yet from eating baboon brains, which were delicious, by the way. And the hunter that killed the baboon in Tanzania, the Hadza hunter, was the one sharing it with me. So it was this very interesting sort of bridging of cultures. Here I am, this westernized human with an iPhone. And here is this, human who's been in the Hadza tribe and is literally wearing an animal skin, handing me this brain, carapace of this baboon. I couldn't say no. Let's talk about dementia. So the biggest thing people think about with dementia is Alzheimer's, which is probably what we'll talk about most today. But how would you frame the discussion around Alzheimer's? I think that the start of this discussion is perhaps a thread that we pull on, saying most of what many people believe about Alzheimer's for the last 15 to 20 years is probably not very useful in treating it or reversing it or talking about how to prevent it, and potentially totally wrong.
That's certainly where I and numerous other people have ended up. That we lumped together two fairly distinct diseases or disease processes and called them all Alzheimer's disease. And that has made figuring out the approach to the more common late onset or sporadic Alzheimer's disease, finding treatments for that, I think has been hindered because we've kind of lumped it in with this other disease, which is the disease originally described by Alois Alzheimer, which is familial monogenic Alzheimer's disease. It onsets in 30s to 50s. It has a very well described homogeneous course. It's much better understood, and we know it has genetic underpinnings, which can either be passed on familially or can the mutations can occur spontaneously, which they do in about 50% of cases. But if people are watching the video, then you can see the table here. This is from our recent paper. The presenilin genes and amyloid precursor protein genes. These are most commonly the genes involved in early onset or classical Alzheimer's disease. And it's thought that the first case of Alzheimer's disease was maybe due to a PSEN1 mutation. There was a paper that did some genotyping of the brain from that patient. It's been disputed since, but it's almost certain that the original description of Alzheimer's disease was due to a single mutation, also autosomal dominant mutation. If you then compare that to what we in this paper called age-related dementia, which is far more common and what people have 95+% of cases of Alzheimer's disease, and what people might call late onset Alzheimer's disease. If there's a genetic component, it's polygenic, very imperfectly penetrant for a number of reasons that we can talk about. Very heterogeneous disease course, very heterogeneous pathology. If we actually look at the brains and see what's going on in there, it looks very different from person to person. And then there's a whole bunch of lifestyle factors that are important as well. It occurs much later in life. So even though if you look at the brains of people with advanced Alzheimer's disease of both types, they may look very similar, what caused that process to sort of end up there is probably very different. And in order to tackle the real burden, which is the late onset Alzheimer's disease, which we called age-related dementia, I think it takes a different approach, and we should sort of just separate it out entirely.
It mirrors some other discussions we've had in the past on podcasts, perhaps these LDL polymorphisms in familial hypercholesterolemia being described as markers or model systems for atherosclerosis. But yeah, there's a difference here between age-related cognitive decline, age-related dementia, Alzheimer's, which is sporadic, heterogeneous course, not related to any one gene that we can think of, perhaps polygenetic. Now, when we're talking about Alzheimer's, I think the gene most people will think of is ApoE. Can we talk about that for a moment? We did a whole podcast on this, but I think that some framing around ApoE 4/4 polymorphism specifically, or 3/4, would be helpful for people from your perspective.
Yeah. So in westernized populations, ApoE4 is having one or more copies of ApoE4 is, the most important single genetic risk factor for age-related dementia, we'll call it. It contributes maybe up to 5% of total risk, your ApoE4, your ApoE genotype. And, compared to those who don't have ApoE4, so either 2/2s or 3/3s or 2/3s, you get an increased odds of developing age-related dementia of maybe 6 to 20 fold higher, which is marked. However, we know that depending on the scenario and the individual, ApoE4 may or may not actually be a risk factor. So in indigenous populations, non-westernized populations, and now we have multiple examples. I think we cited 3 different papers in different populations in that paper that ApoE4 is either not a risk factor or may be protective in certain scenarios. So it seems like ApoE4 is the majority of the risk comes from an interaction between that gene and the modern westernized environment. So when people are not in that environment, it doesn't seem to be a risk factor at all. So as with most things, that have a genetic component, the interaction with the environment really is critical. And you could argue that, maybe we don't know yet which factors in the environment are the most important, but you could imagine a scenario where somebody eats a minimally processed diet, sleeps, exercises, doesn't smoke. Is ApoE4 then, avoids systemic insulin resistance or type 2 diabetes or prediabetes? In that setting, is ApoE4 a risk factor? I would argue probably not, or it would dramatically decrease the likelihood of there being an issue. But sort of, I don't think there are any numbers on that yet because we're still sort of teasing that apart.
So the populations that I remember you and I talked about were the Chimene and perhaps the Bolivian Yoruba. Or is there a third population that?
Yeah, there's a Central African population in Nigeria, and then also indigenous Americans.
So I guess Bolivian, maybe it's the Yoruba. Interesting, because I found that so interesting when we had that conversation a few years ago, that in hunter-gatherer or non-processed food consuming westernized populations, I guess none of these populations are true hunter-gatherers, that ApoE4 did not have an increased risk of age-related cognitive decline or dementia, and perhaps was even protective in people. So I was fascinated to find the history of the ApoE4 alleles and to learn that ApoE4 was the ancestral genotype. Can you talk a little bit about that and perhaps conditions where ApoE4 is even protective?
Yeah, sure. So the ApoE gene, ApoE protein, obviously involved, it's an apolipoprotein, so it's involved in lipid trafficking in the body, but is also associated with immune function and inflammation. And originally, as humans, maybe came down from the trees, for want of a better phrase, as we were exposed to a new and greater number of insults, the original, quote unquote, original human ApoE genotype was ApoE4, and it compared to the others, which came along later, is often associated with a greater or heightened immune response in certain settings. So this is maybe beneficial when you're being exposed to a greater or number or different insults than you may be based on the habitat that you're previously evolved in. And in the setting of the Bolivian Simene in particular, the one study where they looked at ApoE4 genotype and cognitive function, it seemed to be ApoE4 seemed to be protective in the setting of a high parasitic infection burden, which is common in those societies. And that's probably because of improved immune function, able to sort of better respond and mitigate some of the issues that you might get from a systemic parasitic infection. However, in other settings, ApoE4 can still increase risk of worse cognitive outcomes, if you want to put it that way, in the setting of traumatic brain injury, it's like an acute brain injury or a stroke, or maybe even more of a chronic type brain injury, associated with poor systemic health or lifestyle. ApoE4 seems to, accelerate or increase the sort of inflammatory responses into the brain to those kinds of insults. And that's maybe why it can sort of amplify the risk in the sort of the modern environment, which seems to not be particularly good for the brain in general. That's what we're seeing increasingly.
A lot of people get worried about saturated fat when they have ApoE4 alleles, whether they're heterozygous or homozygous. To me, it's never made a whole lot of sense to think that our ancestors were eating a whole bunch of saturated fat from animal foods that they were presumably hunting as they came down from the trees, Homo habilis, Homo erectus, and even precursors, the Australopithecines, and to know that those people exclusively, those hominids and prehominids exclusively expressed ApoE4, because I believe ApoE3 arose around 300,000 years ago, and then ApoE2 maybe 80,000 years ago. I think those are the numbers. And so it's interesting for me to think about it in that context. How do you think about saturated fat consumption from animal foods and ApoE4?
I think in the broader context, I'm not particularly concerned about saturated fat and ApoE4 because I think there are much more important drivers of long-term cognitive decline and cognitive function. However, I do think it's worth bearing in mind that the data that we have from what we think was available for early hunter-gatherer species. If you're thinking about animal fats, the majority of the fat in most wild animals is unsaturated, and the majority of easily accessible animals are relatively lean. So if you compare that to somebody on a modern ketogenic diet slamming heavy cream and butter all day, I think the levels of exposure are dramatically different. So in terms of the saturated fat intake from a minimally processed, potentially varied, doesn't have to be diet, I'm not concerned. If saturated fat makes up the majority of your calorie intake, I think that is a relatively novel exposure, and I don't know how that interacts with ApoE4 genotype. I wouldn't say one way or the other.
Mm-hmm. Now, do you think it's safe to characterize ApoE4 as something that is mostly a risk for people in westernized societies, especially those who are insulin resistant? I see insulin resistance as this hugely important context. You and I have spoken about this on a previous podcast 2 years ago, like swimming in dirty water. What is the context of the people that we are looking at in these populations? And for me, insulin resistance is the main pollutant in that water that I think confounds a lot of what we're seeing, or needs to be carefully considered as a population that is mostly insulin resistant. Do you think it's safe to say that in populations who are insulin resistant, ApoE4 is a problem. And in somebody that's not insulin resistant, ApoE4, maybe not a big deal.
So I think in terms of, with epidemiological observational evidence, I think you could say the former, particularly because in the studies that we have available, large cohort studies in the US or in different countries in Europe, it certainly seems like ApoE4 is a significant risk factor for long-term cognitive decline, age-related dementia. However, we also know that in those populations, prediabetes and diabetes is the norm, is average, right? We think in the US, and now it seems to be reflected in similar studies in Europe, the average person is at least pre-diabetic. So in that setting, I think ApoE4 is a risk factor. If you take that away, does that risk still exist? It's much harder to figure that out because there are just far fewer people to research and try and answer that question. So again, if you look at, so if we go back to the Bolivian Simone, if you look at, they have a 0% risk of type 2 diabetes. Like nobody has diabetes in that population. And they've looked at that. So in that setting, again, we see ApoE4 maybe not associated with risk, but again, it's a very niche, small group. How does that sort of extrapolate to other populations? So my answer is, I don't know, but I do know that at the population level in westernized societies, it is a risk factor. And that comes along with the fact that on average, an adult is pre-diabetic, has at least one chronic condition, takes at least one prescription medication. That's the average. So that's kind of what you're seeing at that population level. My feeling is that once you start to unpick some of those lifestyle factors that we know are associated with increased risk, ApoE4 is going to be less of an issue, but that's just my opinion. I don't have any really strong evidence to support that.
Now, can we just take a slight detour for one moment because this is an important question? I think this is really a super fascinating question within Western medicine. What do you think is driving insulin resistance in our populations? There's been a lot of surveys looking at characteristics of the metabolic syndrome. Some of them show that only 12.2% of the population, and some of them show that even less than that, doesn't have any one of the 5 characteristics of the metabolic syndrome, suggesting that 87 or 88% of people in the US, and I think even more than that in the UK, have, like you said, prediabetes, some leaning toward an insulin resistant state. What's driving that? I think that's a fascinating question that every medical student should be asked, every doctor should know, and that we should really be thinking about. But I'm curious for your perspective here.
Insulin resistance is something that I've been interested in for, a decade or more now. And I think the best sort of cognitive model that I have to explain it is based on body composition and to some extent physical activity, although less so. And when you look at individuals, again, both individually and then at a population level, it seems that you get to this point where you've accumulated so much adipose tissue, and this starts in the subcutaneous adipose, that after that, the subcutaneous adipose tissue becomes insulin resistant, and then you get this knock-on effect of adipose being or fat being deposited in other tissues as and then they become insulin resistant as sort of an excess of energy is stored in those tissues, maybe when it shouldn't be. And what determines the level or the point at which your subcutaneous adipose tissue stores say, okay, I've had enough, I can't store anymore, is probably determined by a whole number of factors. So systemic inflammation. So we know that, say, individuals with autoimmune diseases may be relatively lean, but that large degree of systemic inflammation seems to decrease the point at which the adipose becomes insulin resistant, and they get insulin resistant despite not having much adipose tissue. So systemic inflammation, oxidative stress, probably nutrient status, exposures to environmental toxins, maybe pollution, air pollution, things in the water, systemic and social stresses probably play a role as well. So, each individual has this, point at which they've accumulated adipose tissue beyond the point at which subcutaneous adipose can remain insulin sensitive. And then essentially you're trying to stuff too much energy into those cells. They say, no, thanks, I can't take any more. At that point, they're insulin resistant. And then there's this knock-on effect. I think there is a certain subset of people where, due to the fact that they are completely sedentary, insulin resistant may start in the muscle tissue. Your muscle tissue is by far your biggest glucose sink. 70% of glucose uptake goes into the skeletal muscle, and 75% of that is non-insulin dependent. It basically, that muscle is just there to take up glucose. But if you have low muscle mass or you're not using your muscle tissue, you don't have that sort of capacity for glucose flux. And then you may start to see deposition of intramuscular triglycerides as like the excess energy that's sort of being stuffed into that muscle tissue can't be used. And then the muscle tissue becomes insulin resistant, and the same problem occurs. I think in most people, it's probably adipose related, but in a subset of sedentary individuals, I think that muscle tissue is critically important as well. We also know that if you are active, physically active, that decreases systemic inflammation and probably allows for a greater capacity of adipose storage in the subcutaneous adipose tissue. So you can significantly improve insulin sensitivity with activity without changing body composition, but you have to do it continuously, right? It would have to be an everyday thing. But you certainly can improve your metabolic health without changing body composition by just moving more. So then you might ask, well, what is it that's causing excess adipose tissue to be stored? And in my opinion, there are a number of parts of, again, the modern environment that can drive that, from altered sleep and sleep deprivation, again, chronic and social stress, and then the nature of the food that we eat. And I'm particularly concerned about food processing. So when your food is nutrient poor, calorie dense, particularly when it's soft, and then the combination of all the different flavors. So we have thousands of scientists who are highly skilled in making foods that are perfectly hyper palatable. That sort of milieu is essentially what I think drives a significant amount of overeating, which then leads to sort of long-term weight gain. So that's kind of that's where I see the cognitive model of to explain this currently.
Linoleic acid, Tommy.
Works. It would definitely explains what you see in rodents, but I don't have really good evidence to support that that's what's happening in humans right now, and I don't think anybody does. So mechanistically, could certainly make sense, but I don't think we have the evidence to say that one way or the other.
And you and I have talked about this offline. It's, if we can figure out a way to do that study, I hope we can do it, because I don't know that we have studies in either direction. We have meta-analyses, we have regressions, we have epidemiology, we have murky data points on both sides. And I think it's a really important question that we answer in Western medicine. If that single factor is driving some of this, I wish we could get a better study to do it. But like you said, the time horizon for that study is probably pretty long. It's a very expensive study, and I don't know if it'll ever be done.
Yeah, I don't know either. And I've certainly been very interested in sort of the mechanistic underpinnings of why that might be a significant driver. I'm originally trained as a biochemist. I find it very interesting. But when you're trying to apply this at the sort of the population level, or really sort of intervene, I have to admit that I don't think there's much evidence. And if anything, the evidence we currently have, which again, based on not great nutritional epidemiology at the sort of observational level, if anything, the signal goes in the other direction. But those data really aren't great. So I would say that really we're at a point where it's generally inconclusive. However, I think we do have good evidence to talk about things like food processing and nutrient density and food texture. That's another thing that's become sort of to the forefront. And so then I don't think you need to worry about the individual mechanisms and come up with all these theories about which individual component might be doing it. Because if you address those other things, calorie density, nutrient density, food texture, food processing, which I think you'll overcome most of those issues without having ever having to get into an argument with anybody about linoleic acid, like regardless of what you think about it.
Is there any soybean oil in your house?
No.
Canola?
No. Corn? No.
Is there any seed oil in your house, Tommy Wood?
I don't think. There's olive oil.
That's not it. It's a fruit oil, right?
Yeah, I know.
Okay. It's such an interesting thing, I think, because I see people, and I do these little curious experiments in my head all the time. I live in Costa Rica. It's supposed to be a blue zone. I used to live on the Nicoya Peninsula. It's a blue zone, quote unquote. We'll talk about blue zones here in a moment. A lot of people are overweight, man. A lot of people are overweight. And I see a lot of seed oil everywhere. And this is just a correlation. This is nothing. This holds no water scientifically. But I know that people here eat a lot of processed food. They eat a lot of processed sugar. They eat a lot of seed oil. They don't eat a lot of butter because it's more expensive. They don't eat a lot of tallow. Mm-hmm. And I just wonder about it. I think, what is causing this level of obesity? Because it's striking here in Costa Rica. You do not see many middle-aged men or women who are of a healthy body composition. And I think the numbers or the outlook would be even worse if we drew fasting insulins. So it's just an interesting thing. But I think, to your point, the processing of food, yes. And the other day I was passing by a guy on the street coming home from surfing, and he's got this parrilla, this grill, and he's grilling meat, and he's got this container of seed oil right next to it. And I think, okay, that's not a hyper palatable food. It's not hyper processed. There's no texturization or anything that's been done to it. But here he's cooking meat in what was probably either corn canola or soybean oil. And I think, is that a problem? Is that something people need to worry about or not? I wish we could give people better indications there. I think that, at least from my perspective, and I think you and I have talked about this, correct me if I'm wrong, evolutionarily, our consumption of linoleic acid in 2022 seems fairly inconsistent with what we would have done as humans for the last 350,000 years.
Yeah, on average, it's probably 5 to 10 times higher. I think when they've looked at, again, the closest we can get to is modern hunter-gatherer populations, and it's probably 2 to 3% of calories. And that's why, and I think, again, if you eat generally, you don't eat a ton of processed foods or processed oils, it's unlikely you'll hit that unless it's all soy-fed bacon. So again, I think, if you're concerned about it, you can get down to those levels without having to be, particularly hyper focused on one factor, when I think other factors are going to come into play as well. So that's where I've tended to go. So in your setting, yes, maybe if linoleic acid is the causal problem, and you're basting your meat with it on a grill, I would still argue that if it was just meat with a bit of oil on it, I probably wouldn't worry about it. What else is that? What else is coming as part of that meal? I think you have to take that into account as well.
Yeah, absolutely. It's important and it's interesting, and I don't know that we'll solve this problem in our lifetimes, but
I will have to admit that I've over time, I was very hyper focused on mechanisms which are compelling in rodent data, and I've just had to acknowledge that I don't think the evidence exists in humans. That doesn't mean it's not the case, but I think there are other ways to get at this without having to really worry about whether that's the case or not. And also, I will say, as somebody, my day job is doing basic science work in rodents. I couldn't tell you of the number of times that I've seen something that happens in rodents that doesn't happen in humans. Thousands of times. So I don't find that kind of work compelling anymore.
2,700 calorie diet, high in linoleic acid, 10 to 15% linoleic acid. 2,700 calorie diet, low in linoleic acid, 2 to 3% linoleic acid. This is Tommy Wood's thought experiment. What happens to those two individuals? All other factors equal, 10 to 15 year time horizon.
So if you've optimized everything else, I honestly don't know.
It's hard to say.
I'm not convinced you'd see it. I'm not convinced you'd see a significant effect there. Once you take into account physical activity and sleep and other aspects of the diet and nutrient sufficiency, I don't think we know. So I wouldn't hazard a guess.
Okay. I want to touch on this and then we'll get back to Alzheimer's and dementia. Calories in, calories out. That model for weight loss. Your thoughts on that? Your thoughts on that, Tommy?
I think the current iteration of the energy-based model as has, there's been this people are probably aware there's been this back and forth between David Legowig and Kevin Hall and their various cronies over the two, sort of prevailing models of energy metabolism and obesity. I think the current iteration of the energy-based model from Kevin Hall is the one that's closest to the truth. I think, yes, calories in and calories out are an important factor, but you also have to take into account that calories from different foods, from different macronutrients, affect the energy out in different ways by affecting physiology and satiety and physical and non-exercise activity thermogenesis, all that kind of stuff. So I think that calories matter, but you have to take into account how different calories from different sources affect the whole system and affect physiology. And I think the most modern versions of that model do that. So that's where if I had to stake my claim, that's where I would stake it.
Can you expand on that a little bit? Just so people understand what you're talking about. Are you talking about macronutrients? Are you talking about different types of fat? Because I think that at least in rodent models, like we talked about, there's some concern that certain types of fat may affect satiety differently. I guess you could start with macronutrients, and then if you want to break it down within macronutrient categories, I'm interested in that too.
So I think macronutrients is definitely the best place to start. So the things like we know, quote unquote, know that protein is probably the most satiating of the macronutrients. That also has a higher thermic effect. So even though it technically has the same number of calories per gram as carbohydrates, that doesn't really translate into the same number of calories that's sort of like available for utilization by the body because of the thermic effect. And then it is probably more satiating. So individual macronutrients can have different effects on satiety. We also know that so everybody talks about the fact that a plain boiled potato is the most satiating food of any that's ever been tested because it's hard to eat and it's boring to eat. So, right, how many boiled potatoes with no salt or anything on could any one individual eat in one setting? Not very many, right? So they are quite satiating. So it depends on both the macronutrient, but then also the food context, and then how that food is seasoned and flavors and all that kind of stuff as well. In terms of within macronutrients, say there are some data to suggest that let's focus on carbohydrates. If you process carbohydrates that on average, you can't. Nowadays, we know that any individual, you can't predict how they'll respond in terms of their blood sugar to a certain carbohydrate containing food. But on average, if you sort of process, so say you take a grain and you grind it down and you boil it and then you eat it compared to eating that sort of like whole grain cooked, the true whole grain, you get a much larger blood sugar spike from the former, sorry. And so when you process food, you start to dissociate the macronutrient and calorie content from the normal physiological responses that we may have elicited when we were eating that food in its minimally processed form hundreds or thousands of years ago. And if you have a large blood sugar spike, it's more likely to lead to a rapid drop of blood sugar later on. And then the speed of that drop and then the degree that it drops seems to then drive hunger later on. So if you eat if you cause very large swings in blood sugar, that seems to increase hunger earlier later on for the same number of calories. And there are some recent studies with continuous blood sugar monitoring that show that. That's probably just like a small effect, but these things kind of add up. In terms of, say, different fats and how they affect satiety, I don't think we have really good evidence in humans. Again, we can go to mechanistic data, the F to N ratio, so FADH2 to NADH production, which is different depending on the saturation level of the fat. That affects how it affects reactive oxygen species signaling in the mitochondria that made feedback to the insulin receptor, which can then affect satiety again. And this very mechanistic seems to explain what we see in mice. When we do overfeeding studies in humans, where we very specifically make the fat either polyunsaturated linoleic acid or saturated fat, we don't seem to see that difference. However, it's been done in the context of a mixed diet. So, so like the muffin study in Sweden, So it's the fat in a muffin. If it was pure, I think it was either palmitic acid or linoleic acid. It didn't seem to have an effect either on satiety or the amount that people ate. But that's in the context of a mixed diet. If you were sort of isolating it even more, there's a possibility of an effect, but I don't think we've really seen a strong signal in humans in the studies that have been done so far.
Have you seen the study? It's in one of the Nature journals with stearic acid, where they had vegans and they deprived them, or they had people eat a vegan diet for a few days. They deprived them of any stearic acid. Then they gave them a shake with a large amount of stearic acid and saw changes in mitochondrial function, or at least fusion. And they saw changes in beta oxidation markers. Have you seen that one with stearic acid? I thought that one was particularly interesting.
Yeah, I think so. I read it when it came out. Was it a couple of years ago now? 2 or 3 years ago?
I think so.
I haven't read it since then, but I do remember reading it.
I just found it interesting that stearic acid had that specific effect. And I think that what I'm curious about is just like we're talking within macronutrient groups, especially within the fats, are there certain fats that have pseudo hormonal effects in the human body and can affect things differently than other fats? Assuming that we're at 9 calories per gram for all of them, that's an interesting question for me.
Yeah, and I mean, it is possible, but you have to remember that these sort of short-term mechanistic outcome studies don't necessarily tell you anything about how things integrate long-term and then result in differential satiety, total calorie intake, body composition, long-term health outcomes. So again, this stuff is interesting, but until we have a really good way to either randomize or assess different levels of stearic acid intake within individuals who are otherwise similar to one another, I'm not really sure I could say one way or the other.
We touched on blood sugar. How do you think about that? Because there are many in the health community who get very worried about absolute levels of blood glucose. You and I have talked about glucose excursions from the mean in the past and blood glucose variability, other metrics. How do you think about blood sugar? I think you've probably worn a CGM in the past. Do you worry about absolute levels of blood sugar spikes? Do you believe there's a ceiling that we should not exceed for blood sugar? How do you think about this?
So I think blood sugar can, large spikes and frequent spikes in blood sugar can have an impact on long-term health. There are some studies, again, sort of associational, but looking at MAGE, so mean amplitude of glycemic excursions, and degree of atherosclerosis, risk of diabetes. I think there's a signal there. We don't understand it perfectly, but there's also there's some data to suggest that very large and persistently elevated glucose may affect endothelial function, which then could start to contribute to the process of atherosclerosis. May also affect blood-brain barrier function, may start to affect cells within the brain. So I think there's some potential for an issue there. However, again, I think if we focus on some of the things that we mentioned earlier, body composition, movement, diet quality, I think a lot of that starts to fall out. CGM can be useful, but what I'm really concerned about now in sort of like the health focused communities is this: we've essentially we're pathologizing food, with a particular focus on blood sugar, because that's what we can measure, and you can kind of see it, right? So you have your favorite scientific or physician influencer on Instagram, and they're wearing their blood sugar monitor, and they're telling you about this highly processed food that they're about to eat, and they're already stressed about the blood sugar response that it's going to cause, and they've essentially created this like pathologic association between certain foods and their health. And I would guess that the stress of worrying about that is probably worse than the blood sugar spike that you get from that meal, assuming that it's happening intermittently. So what I'm seeing is what looks like disordered eating driven by blood sugar. We also know that expectation drives blood sugar. So if you think you're going to have a bigger blood sugar spike because you ate more carbohydrate, you will have a bigger blood sugar spike. There are studies done in diabetics by Ellen Langer that have shown this, randomized placebo-controlled studies. So if you're wearing a CGM and you think this food is going to spike your blood sugar, you'll get a bigger blood sugar spike because you're expecting it. So if you think blood sugar spikes are pathologic, then you're almost creating one for yourself as part of that process. And I think that's a potential problem. If you're somebody who's in a position to use a CGM and you think you could get useful data, I would argue that you probably only need to use it for 2 to 4 weeks max. And that's because most people eat the same thing again and again and again and again. And how you respond to individual foods is fairly repeatable. So if you wanted to know, hey, how much does my blood sugar spike when I eat my local Costa Rican papaya? How many times do you need to do that before you know, right? Probably 2 or 3 times, and that's it. So I would argue that you could probably collect all the data and information you need in 2 to 4 weeks, and then you never need to look at a CGM again. And maybe it is, well, hey, bananas don't spike my blood sugar a lot, but papayas do. So maybe you just eat fewer papayas, and you just know that. And there's a possibility that as your health changes, your body composition changes, you get older, some of these responses may change. So maybe in 2 or 3 years' time, you need to do it again. We don't know yet. We also, if we're honest, we don't have any good prospective data that tells us how well, blood sugar variability predicts long-term health outcomes. What we have is, if we look at your blood sugar variability right now, how does that associate with your current health? That's very different. So we don't know if we change thing, or there are only a small number of studies, and I can talk about one. But we don't know if you sort of like intervene, specifically focused on glucose, does that have a meaningful effect on your long-term health? We just don't know that. The one study where I have seen that done, and there may be others, is in a Japanese study looking at dipeptidyl peptidase 4 inhibitors in type 2 diabetics. So these drugs basically prevent the breakdown of incretins. So you get improved blood sugar regulation. It's just, a similar effect to, the GLP-1, GIP agonists that are now really popular, like semaglutide and tirzepatide. And in this study, they showed that, and it was in older individuals, basically your mean amplitude of glycemic excursions was your best predictor of your current cognitive function, as measured on a mini mental state exam. And then 2 years later, after taking these DPP-4 inhibitor drugs, the more your glycemic excursions improved, so the spikes got smaller, the more your cognitive function improved. So this is in older individuals. They've improved their glycemic excursions, and then that's associated with improvement in cognitive function. I think that's some nice evidence to suggest that maybe there's a link there. But of course, along with those improvements in glycemic excursions, you've also got improved, weight loss, improved body composition, all that kind of stuff, which plays a role too. But that's probably the one study where I think they've looked at it longitudinally and then looked at how that's associated with differences in various health metrics.
Given someone like myself with a fasting insulin consistently below 3 micro IU per ml, do you think I need to worry about a postprandial blood sugar of 150 milligrams per deciliter?
No.
Okay. Simple answer. 150 is that gives physician influencers who are focused on glucose a heart attack, I think.
I think, in somebody who's very healthy, if your fasting blood sugar is between 80 and 95, maybe even 100, and your MAGE is less than 50, right? So maybe you're going from 100 to 150. I'm not convinced that's going to be pathologic.
And just so people know, that MAGE is the glucose excursion. So it would be the amount that your glucose moves from your average baseline to the apex of the postprandial spike in the glucose.
Yeah. So if you think about each individual spike, and then across the day, and then you would average those spikes to get the mean amplitude, so the mean height of the spike. So say you eat 3 meals a day, each you go from whatever your baseline is, it goes up by 50 each time, then your MAGE would be 50. But if it was like it went up 50 and then 40 and 30, then your MAGE would be 40, which is the average of the three.
Okay, 40 is the average.
In that specific scenario.
Right. And if somebody wants to look at the MAGE, say they have a CGM, do you have a metric that you use, a 50 or something like that?
Yeah. So if I thought that you were high risk, and there's some data to suggest this, then I think that you should target a MAGE less than 40, maybe less than 30. If you're very low risk and everything else looks great, and you have a CAC score of 0, and good body composition and sleep well and all that all that kind of stuff, then I would suggest maybe a limit of 50 or 60 would probably be fine.
Yeah, I haven't done my CAC for 2 years, but it was 0 2 years ago. I think I don't know what my body fat is. I think that I've got a scale here. It tells me it's 13%. Who knows if that's accurate or not? I can see my abs. That's enough for me.
You spend so much time naked on Instagram that I could tell you that it's probably less than that.
Less than 13. What is the Tommy Wood estimate of my body fat based on my shirtless photos on Instagram?
It's probably, I guess closer to 10.
All right, 10. So that's good. My fasting insulin is 3. So that's probably where it is. And historically for me, for context, I added carbohydrates back to my diet. This is a conversation that you and I had many years ago, because when I was strict keto for a long time, I had numerous issues. And now I find that with a few hours of exercise per day, that being paddling sometimes for my life in the ocean, sometimes it feels like that. More carbohydrates seem to do well for me, and I don't see anything going in the wrong direction in terms of inflammation, in terms of average blood sugars, in terms of fasting insulin, et cetera, based on my recent labs. So that's helpful. Thank you for taking that little detour with me, Tommy. Let's go back to dementia. So we branched off. We talked about ApoE4, and we were sort of talking about how the model for Alzheimer's dementia has been wrong. So we talked about ApoE4, but bring us back into that and talk about how we perhaps have conceptualized age-related cognitive decline, Alzheimer's dementia, that is not this, the early onset Alzheimer's. We differentiated those in the beginning, and then how we might create a better model for what's going on here.
Sure. So because of the original way that we understood Alzheimer's disease, and the way that neuroscience really started was with pathology, neuropathology, right? So you take a section of brain, you put it under a microscope, and you take a look at it. And what we saw in classical Alzheimer's, and what you do see in multiple different neurodegenerative conditions, including age-related dementias, is these like pathological accretion of proteins. Like people have heard of amyloid plaques, tau neurofibrillary tangles. So these are two proteins that misfold or aggregate within the brain that can either be outside of cells, as you generally see with amyloid, or inside of cells, as you generally see with tau. And there is a point, and we see this in human data and then also in animal data. There is a point where once you've collected enough of these proteins, they start to be sort of damaging in their own right in a feed-forward manner. However, there's this like gulf in between where different people have vastly different amounts of these proteins, and they have very different cognitive outcomes, or long-term cognitive trajectories, and the two almost don't correlate at all. So once you get to the end stage of the disease, yes, these are certainly involved, but it seems like in between they don't really make much of a difference. And we've seen this in multiple mouse models, that the amount of amyloid that you've accumulated doesn't correlate with outcomes. We see the same thing in humans. In humans, we can actively reduce the plaque burden, the amyloid plaque burden in your brain, but it doesn't seem to have any effect on your cognitive outcomes. And this is where billions of dollars of amyloid targeting therapies have gone by the wayside. And I've heard from some people that they think there's going to be a new study with aducanumab, which is going to show that all the naysayers are wrong, and actually it's going to be beneficial. But in general, there's no therapy targeting amyloid has been shown to be beneficial, and that amyloid plaque burden doesn't correlate with cognitive outcomes or trajectory of Alzheimer's disease. So then we started to focus more on tau, which may be a better predictor, and people are now talking about a dual pathway model where these things sort of like come together and interact. Other people are talking about something like a probabilistic model. So they're starting to take into account other factors. So then genetics becomes a factor, other environmental components become a factor. So we know sleep is important. We know that toxic exposure like air pollution is important. We know systemic health is important, like hormonal status, particularly in women during and after the menopausal transition. That's a significant risk factor. So we know all these things are important. And now, sort of the state of the art is to say, well, we can kind of, as all these things come together, the probability that you get dementia is increased. So, based on your hormonal status and maybe nutrient status, also very important. So some, really critical studies done by David Smith, who's an emeritus professor at Oxford. The Optima and VitaCog studies basically showed that if you have elevated homocysteine, so a level above 13, and you reduce that by giving somebody B vitamins, so particularly B12, folate, B2, and they have adequate omega-3 status, there seems to be an interaction, then you can significantly slow brain atrophy and slow cognitive decline. So nutrient status certainly seems to play an important role as well. And so at the moment, we're kind of like, well, all these things are important, and you'll have seen some models where say the work of Dale Bredesen, which again has just been sort of fairly groundbreaking, and he's shown that in people with various stages of cognitive decline, you can reverse it by addressing multiple risk factors at the same time. So systemic inflammation, nutrient status, physical activity, diet, sleep, stress, all this kind of stuff. But it's a fairly complex process and very difficult to scale. You basically have to assess 50 different things at a cost of a vast amount of money and try and figure out, well, what's the thing that I should fix in this individual? And there is this still sort of very heterogeneous process that's going on. And all of that will be important if you want to like absolutely optimize or maximally minimize your risk of cognitive decline. Where we're going with this is the paper that Josh Turknett and I recently published, which basically sort of completely changes where we focus our efforts. And so all of the stuff that I just mentioned, I think is important. I don't want to discount it because I do think it's important. We have sort of interventional studies that show that, if we change stress or physical activity or nutrient status, we can reverse some aspects of cognitive decline or improve cognitive function in older individuals. But we start the paper by arguing that we're looking at the wrong biological level. Like we're hyper-focused on proteins and mechanisms and in the cells, when what's probably most important is happening at like the organismal societal level. And what we suggest is maybe the most important both risk factor and then potential sort of mitigator of long-term cognitive decline is how and how much you use your brain. And so we liken it to exercise. And there's lots of evidence that actually shows that the way the brain responds to increased cognitive demands, and by that I mean, skill building, language development, sort of complex brain training, rather than I'm busy all day, my brain is busy. Those are not the same thing. But developing skills, that kind of cognitive demand, the cellular responses are very similar to how the muscles respond to exercise. And they respond by getting stronger and more resilient with more connections, and muscles do the same. And so the way to kind of boil it down is to say that, so imagine you, Costa Rica, great lifestyle, great food environment, no stress, not much pollution, right? You've optimized everything and you want to get stronger, right? You can eat all the protein you want, but you're not going to get bigger and stronger unless you actually stress your muscles, right? Mechanical tension is like king of increasing muscle strength. And I think the brain is the same thing, right? You could have this pristine environment where, even with, so movement helps. So, any kind of movement. So even though you're moving, you eat a good diet, you're not stressed, you sleep. How are you going to maintain the strength of your brain if you don't challenge it? I think the sort of the analogy works very well. So like a primary driver being how much are you really tasking your brain and asking it to adapt and be plastic and get stronger in terms of its connections and its resiliency. And the thing is that most individuals in modern society don't do that, right? We do the same thing every day. Our work isn't cognitively challenging. It's repetitive. Yes. It's stressful. Yes. But those are not the same thing. And so, we're not developing new skills. We're not asking our brain to do new things because we don't like being bad at stuff, right? Human adults in Western society just cannot. They just hate being bad at something and being seen to be bad at something. Yet that process of learning and failing is probably what is most critical for keeping a healthy brain. And I liken it to the brain developing in the first place. When you're a baby trying to develop your brain, you are constantly trying new things and you're constantly failing and falling over and not saying the words properly and interacting poorly and falling out of trees and all that kind of stuff. But as a result of that, you develop incredible motor skills, incredible language skills, incredible social skills. But we don't challenge ourselves in that same way as we get older. And with that, you get this trajectory of decreased cognitive function. And the easiest ways to see it is that when we retire, now there are multiple population studies that show that the earlier you retire, the earlier you experience cognitive decline or dementia. And that's because, in modern society, your work is probably the place where you're most cognitively challenged, even if it's not that challenging relatively. And as soon as you remove that, you're not going and then traveling and learning languages or learning to slackline or doing yoga, or at least most people aren't. You just go home and sit and watch TV, and your brain just's like, well, I'm not needing it anymore, so I may as well self-destruct. And there are molecular pathways by which that happens. So that's kind of what we've proposed, that the most significant protector of long-term cognitive function is how much you use your brain.
And I appreciate the way you differentiate between just being busy and doing something that's stressful and that occupies your time and doing something that really challenges your brain. When I was in medical school, I started learning jujitsu, and it was horrible. It was horrible. I hated it and I loved it at the same time. I never got that good, but I did it for a few years and I enjoyed it. And eventually my body learned how to move a little bit in those positions. But I could feel there was something going on in my brain. Sometimes after training or drilling, I would feel that my brain was sort of sore. It's like the Gary Larson cartoon, may I be excused, my brain is full. And then I did a little bit of training in stand-up martial arts like Muay Thai. I also never got very good at that. And I felt the same way. Sometimes working on boxing combinations or something, I just couldn't get it. But I feel my brain doing that. The same thing probably happened with slackline earlier in my life. And I'm thinking about myself, and I'm thinking, okay, now I surf every day, which helps me a lot because it's super challenging and I'm constantly trying to do that. And I live in Costa Rica, which has challenged me to be more and more bilingual. And in your paper, you mentioned that bilingualism is protective against cognitive decline. I thought, oh, this is great. I mean, my Spanish isn't perfect, but it's gotten a lot better being here. Mm-hmm. And languages feel the same way to me. And you mentioned that in your explanation there, that it feels like you have to try and fail. But you're right. I don't like being bad at things. It sucks, dude. I hate being bad at surfing, and I'm moderately good at surfing. I'm not great. But it's making me think that I need to constantly be doing new things. That okay, I can challenge myself with surfing every day, but like, what am I going to learn next? Maybe it's more Spanish. Maybe it's another language. Is this how you would challenge people to think about things? Like, what are you actually learning now?
Yeah, exactly. And the important part of that being, then, once you're good at something, you have to do something else, right? Because once something becomes habit or rote, or you can do it automatically, it's no longer a cognitive challenge. So I think one thing that most people will relate to, which is ironic because I don't do it, but is driving. It's hard. When you first learn to drive, it's difficult. It requires all parts of your brain, and you're continuously thinking, and everything requires input in order to do it. But then later on, it becomes automatic. So once it's automatic, it's no longer a challenge, and then you have to do something else, right? And then, but what I think is good, there is some specificity, right? So if you particularly want to improve the strength, the performance of your hippocampus or your memory related parts of the brain, then memory related tasks do have a greater benefit there. But in general, I think you can think about, well, what's a skill that I'd like to learn? And whatever it is you enjoy is probably something that you could do. So it could be knitting, or it could be yoga, or it could be a language. Maybe it's novel social interactions, right? Because for some people that can be a stressful or a skill to develop. So there's a whole bunch of stuff that you could do, and anything that you can spend sort of concerted time learning, but then requires a recovery period is where you think, well, this is probably the kind of thing that I need to do. So if it's something that you could do endlessly for hours and hours, it's probably not at the right level of challenge, right? If you think about learning a language, learning a new movement, some kind of coordinative type skill, you can probably only do it for 20 to 30 minutes in like a really concerted effort. Right. So if you think about you're doing your jiu-jitsu level, your lesson or class, you do some warm up and some patterns and that kind of stuff, but you probably only spend maybe 20 to 30 minutes to like really focusing on new skills, right? Because that's probably the time during which the human brain can probably have the capacity to just like really sort of push the edge and fail and get frustrated and all that kind of stuff. So those are the kinds of skills, but you could apply it to anything. So for me, I have spent a lot of time learning how to code. It's important for my job. It's like learning a new language. I'm not very good at it. It's very frustrating. But that's, again, a sort of a modern skill that I think counts. Yesterday, I was interacting with somebody on a post about this paper, and they asked about video games. I don't think we have great evidence for video games because we're only right at the beginning of the generations that grew up with video games getting to the point at which they sort of start to be at risk of cognitive decline. But, you have motor skills, reaction time, spatial memory, problem solving, right? A video game might give us a lot of those things. Of course, you might be sedentary for 8 hours at a time if you're
that's not good for your insulin resistance.
Right. If you're a really dedicated gamer, yeah, there are other things to take into account. But I think all of these things are relevant. How are you really challenging your brain? And there's multiple different ways to skin that cat. So you should just focus on something that you enjoy. But once you're really good at it, you have to try something else.
In the paper, you mentioned the blue zones. I was living on the Nicoya Peninsula, which is a supposed blue zone. People can refer back to our previous podcast about why the blue zones are kind of BS. But nevertheless, I think that there are some takeaways from the blue zones. One of the ones you point out is not the diet of the blue zones, which we know has been mischaracterized, or I think has been mischaracterized. I think you would agree with that, considering they eat meat in most of all the blue zones, except for perhaps Loma Linda, where they have hypoactive sperm. But they don't retire.
Based on the 3 people that they measured in that one study.
Yeah. Well, the vegetarians were low too. So we need a bigger experiment about Loma Linda sperm, is what you're saying.
Yeah, fine. You can volunteer to go and collect samples.
Well, I mean, mine won't be any different. I eat a lot of meat. I'm presuming mine work just great. Like you mentioned that in the blue zones, people don't retire. And I definitely see that here. I see people who work into old age. It's not a culture where the goal is to retire and sit on a beach. You support your family. And so into your 50s, your 60s, maybe even your early 70s, as long as you're functionally able to, there are men doing physical labor and women doing things that women do here, which is usually some sort of like selling things or cooking or, making things. It's just the societal way here. But I do see that here. And I think it's interesting that there isn't this retirement. It makes me a little worried for my family specifically because my dad is now 72 and he's retired from medicine. And he has my niece and nephew. He has his grandchildren that probably keep him active and engaged, perhaps a little bit doing things, but it does make me, retirement is probably a really bad thing for humans, right?
I think so, as long as you don't, replace it with something else that's similarly engaging. Like, again, we, in the paper, we draw an analogy to the sarcopenia or the loss of muscle mass you see in the setting of being either like bed rest, or say you break a muscle or you break a leg and you put your leg in a cast, right? You don't move that leg for several weeks, and then the calf muscle on that side gets smaller, right? Because you haven't used it. And again, all the data support that if you stop using the brain, the same thing happens, right? Metabolic resources are redistributed. You don't need to support those same connections in the brain because they're not being used, and you'll start to pare back. And so you'll end up with something that we call cognitive frailty, which basically means that your capacity for cognitive function is only as much, is basically the same as you use it every day. There's no headroom, right? There's no capacity to do more if you wanted to. And then eventually you get so little capacity that you can do less, and then doing less decreases capacity, and you get this sort of feed forward cycle. So if you were to replace work with something that's similarly cognitively engaging, and it could just be that, right, you retire, but you take up learning Spanish and doing yoga, that's fine. I think that's probably going to do the trick. But again, in general, Somebody might be like, oh, well, I retired, but I do the Sudoku every morning. So that's it. I'm probably good. That's probably not enough. It's maybe better than nothing, but it's probably not enough. So as long as you remain engaged, I think you will get, you don't have to worry about retiring, but you just have to be very, you have to be very cognizant and about that. If you can be, if your brain is still working.
I think Jordan Peterson has talked about the fallacy of sort of the sitting on the beach with the Mai Tai narrative that most of us grow up, at least myself and the culture that I grew up in, thinking, oh, that's when you've made it, when you can just sit on the beach and relax when you're retired. But like, what the heck are you going to do? That's going to get boring so fast.
My wife and I spent a week on the beach with Mai Tais earlier this year, and we may do it every year, but a week is enough. After that, I'm ready to do something else.
What beach were you at?
We were in Maui.
And you just drank Mai Tais for a week?
Something like that. 5 days. Yeah.
I can't imagine Tommy Wood just sitting around drinking Mai Tais for 5 days. You lifted weights. You did something else.
Oh, yeah. Oh, yeah. The hotel had a great gym.
But it was just relaxation.
Oh, yeah. Yeah. And sunshine.
Okay. Sunshine, of course, because Tommy lives in Seattle, which is not as, shall we say, solar radiation gifted as Costa Rica.
I mean, it's okay for like the 3 months of summer, but yeah, you're right. The rest of the time, it's not great.
So I had my vitamin D checked recently, and I'm pretty proud of my 75. 75. Although on a recent YouTube video, somebody said I looked sunburned, and I was like, okay, look, it's just I'm really tan. Okay, I surf all the time. So I think that this is really interesting, and especially on the heels of really, I would say, the failure or the reimagining of a paradigm for dementia, which cannot be based on drug targets for amyloid plaques or tau fibrillary tangles, as far as we know now. Doing something that makes your brain hurt is critical. And we've talked about what those things are. Do you want to talk a little bit about nutrients as we wrap this one up? I mean, what nutrients are critical for brain health from your perspective?
Sure. So I think, and again, it's really funny. So we sent this paper around a lot. So first, before we published it, we sent it to like people we trusted to give an opinion, but who know the field well. And then it goes through peer review. And so we get some opinions from the peer reviewers. And then after it's published, we send it to some other people to say, hey, we published this paper. Like, what do you think about it? Because we obviously have this idea, but it doesn't become really meaningful until like people have kicked the tires on it. Maybe we've updated the constructs, like the model, better understood it, taken more things into account, right? This is by no means like the final answer, of course. And then you create some kind of model or theory. Ideally, we'd want to test it in some way experimentally. But every person who you send it to has a different favorite thing that they're like, oh, but what about this? This is also just as important. And so all these things are important. And I think the way I think about it is that the demands, the critical, like the challenge is like the most important thing. Everything else can be perfect, but if you don't have that, I think you can still end up with a decrease in function. But in order to respond to a demand, you need three things. One is adequate, nutrients and say vascular supply. So you need good vascular health to provide enough oxygen and nutrients. And we know that if you have chronic vascular disease, that can sort of cause atrophy in the brain just because you can't respond to cognitive challenge because you can't. This is a thing called neurovascular coupling, which is where the neurons are more active, so they ask for more blood supply. But if you can't supply that because you have a vascular disease, right, then that's a problem. So good vascular health is important to actually supply the things you need. And then having the things that you need in terms of nutrients. You also want the absence of toxins that might prevent your ability to adapt. So that's where we think about pollution, maybe heavy metals, maybe infections and inflammation. Right, so you want all the nutrients and the supply. You want the absence of sort of toxic exposures, and then you also need time to rest and recover. So that's where sleep is really critical for dementia, and then also the absence of like a chronic stressor that doesn't allow you to really recover. So I think all of those things are required in order to adequately respond to the demand. there are a few studies that suggest that even in the setting of those things not being ideal, you will still get some benefit from demand, which is why I think it sort of has this prime place. The same thing being that, right, if you may not optimize your lifestyle and your sleep and your protein intake, but you'll still get stronger if you go to the gym, right? You could improve that, right? You could optimize that, but going to the gym will still be beneficial to some extent. So in terms of the nutrients, like you asked, I think we have the best evidence for the B vitamins. So there's probably something related to methylation, the one carbon cycle. So B12, folate, B2, really important for lowering homocysteine, particularly in people with certain SNPs.
Which is riboflavin.
Riboflavin, yeah.
Yeah.
And then omega-3 status is important as well. And for a long time, people thought that like DHA was going to, the long chain omega-3 fatty acid was going to be like this panacea for Alzheimer's disease. And it hasn't turned out to be, probably because it's interacting with a few things, including B vitamin status and maybe insulin sensitivity. But those are the things that I think we have the best evidence for. So B vitamin status and then omega-3 status. So in the interventional trials, the best outcomes came from those who replaced their B vitamins or taking adequate B vitamins and had adequate omega-3 status. I think it was like the top third of people based on their omega-3 status. So there's some interacting effects, and those are the nutrients that I think we have the best evidence for.
So kind of bringing it full circle to our discussion of desiccated brain earlier in the podcast, and the fact that when people looked at phosphatidylserine derived from lecithin from plant sources, it didn't seem to have the same effects. I always am a fan of the whole foods. Where can people get those nutrients from the whole food source, with sort of the assumption that there may also be other beneficial things in those foods?
Animal products and organs. That's the answer you wanted, isn't it?
Well, you can say whatever you want, Tommy. You know that.
I think.
When have you coddled? When have you given me the answer that I want?
Yeah, almost never. So, seafood, meat, and organs, essentially.
I mean, I think that's important to point out that when I look, and correct me if you see this differently, when I look, there's no place to get vitamin B12 other than animal foods. End of story, full stop.
Yeah, I think the smartest people in the plant-based arena are recommending methylated B vitamins because you just can't get them from plants. Cyanocobalamin is not really a B12 replacement. And
yet it's in 98% of products, synthetic products that say they have B12, say cyanocobalamin rather than adenosylcobalamin, methylcobalamin, etc. Riboflavin is another one, Tommy. It's tough to get the RDA for riboflavin unless you eat a moderate amount of meat and some heart and some liver, as far as I can tell.
Yeah.
Do you?
And
is
again,
that a reasonable?
I think that's reasonable. And again, I don't have a strong opinion. If people, for various reasons, want to eat a plant-based diet, I would just make sure to supplement that. All of those. There's the focus on B12, but I think riboflavin, some of the other B vitamins, and long-chain omega-3 fatty acids also important.
Which don't really occur in plants either. We can point that out, that like DHA and EPA not really found in plants. ALA, yep. Alpha-linolenic acid, perhaps, but the conversion is variable in different people and may depend on your omega-6 consumption. Would you say that's accurate?
That's accurate. And yeah, it depends on genetics to a certain extent. I'm not a big fan of nutrigenomics. I think most of it's complete nonsense. But there are some data to suggest that your most recent ancestry and where your most recent ancestors, say the last 1,000 years, got the majority of their polyunsaturated fats from may determine whether you'll be better off with the shorter chain or the longer chain version. So if your ancestors came from near the equator, they were agriculturalists earlier, then they probably got omega-3s and 6s from plant-based sources. You're probably a bit better at elongating those and to create your own EPA and DHA. But if you're, say, of Northern European ancestry like I am, my ancestors were eating fish And so they probably never developed the ability to turn ALA into EPA and DHA, or at least very inefficiently. So for somebody like me, it's probably better to get it from seafood.
And all of my listeners will know that I do have a strong opinion. You should definitely not do a plant-based diet. That's a nightmare. We'll agree to disagree on that.
Fine.
But no bueno. Come on. What'd you have for breakfast this morning, Tommy Wood?
What did I have? I had chicken salad that I made the other day, but that's yogurt, some apples, some chicken, and some mustard and lemon juice and some stuff like that.
Okay. What are you going to have for dinner?
Actually, I don't know yet. That's a good question. Maybe steak.
Steak. There you go.
And some vegetables that we grew in our garden.
What vegetables?
I think we have some squash in the fridge and some
That's
tomatoes.
A fruit.
All right, fine. And tomatoes, also a fruit.
Tomatoes are
a nightshade.
Oh no, you're not a fan of that. Could be a problem. Just saying. Who knows? Maybe you're okay. You may not die. You might die. You might not die. Tommy, thanks for coming on the podcast. Anything else you want to say to tie it all in a bow? Anything we didn't get to that is burning in those Viking guts of yours?
No, I don't think so. I think that was a good summary of loads of interesting conversations that you and I have had sort of in the background over the past couple of years. So it was really nice to get those out.
We'll do another podcast talking about LDL soon. Thanks for coming on, brother.
Thanks so much.